So now I take over. It's a great pleasure for me to present the next speaker. And a very good friend of mine, Marta Espinosa. So Marta Espinosa graduated as a surgeon from the Faculty of Medicine of the National Autonomous University of Mexico and obtained a PhD in science from the Center for Research and Advanced Studies in Mexico City in the specialty of experimental parasitology. Marta has done a postdoctoral fellowship at the Rockefeller Universities and the New York University and has been a visiting research fellow at the National Institute of Health in Hester, Maryland. And most recently, she was also a visiting researcher at the Human Genome Center of the Institute of Medical Science of the University of Tokyo. And Dr. Espinosa has been distinguished with several awards, so including the Lola and Biología Award from the National Autonomous University of Mexico for the postdoctoral thesis, as well as the Miguel Alemán Award of Health Science for the achievement as a young researcher. And currently she's working on two major topics. First, immunopathology of multiple sclerosis, where she collaborates with the National Institute of Neurology and Neurosurgery, the Japan Foundation for Cancer Research, and the University of Tokyo. And she developed a three-dimensional human models for the study of intestinal infectious diseases in collaboration with several other institutions. And since 2020, she has now the position of Clinic Secretary at CSF. And at the end, she will talk today about the story of two amoeba.
Thank you very much, Iris. We chose this topic because next year will mark 100 years of a very important observation in the field of amoebiasis. And this is why we, Adolfo and I, said let's do the tale of two amoebas. So our story begins here in the University of Paris in the Faculty of Medicine with a French physician, Dr. Emile Bront, that in 1925 made a very big assumption that had to wait many years to be continued. So here is Dr. Bront. He is in this picture. He is sitting at the lab in the Hospital General de Mexico in Mexico City in 1932. But let's go back to his assumption in 1925. So he said that Entamoeba histolytica that had been already described many years ago, and so was the causative agent of amoebic dysentery. But that there was a second amoeba, which he called Entamoeba dispar, that he claimed was present as an asymptomatic carriers and was capable of invading the colonic mucosa. So after he based his assumption on many studies, of which one of them was that he was able to identify the infection of Entamoeba histolytica in animal models, producing these big ulcers that are also present in their patients. Why infection from asymptomatic carriers that he isolated did not produce any invasion? So this was done around 1933 in his lab at the Faculty of Medicine in Paris. And it is nice that we at CSF have one of his original slides from which these two pictures were taken where he was trying the animal model in cats. So these are original images. The images of the original slides of Emile Bront. So, as I said before, for many years, nearly 50 years, nothing happened. It was his assumption, but nothing really went on. Nobody dared to say anything additional to Brooks' assumption until Adolfo Martinez Palomo made a discovery. He was studying cancer cells, and he was studying the surface properties of cancer cells, and he made a very interesting observation. He saw that those parasites that were isolated from patients with amoebic dysentery were able to agglutinate in a very, very impressive form when put together with concanavalin A. He had used concanavalin A in his studies of cancer cells and had proven that the surface of these cells also have very specific characteristics. But I am not going to go into that in this moment. But the difference was so impressive, like the ones you can see here. This is an isolate from an amoebic dysentery patient that agglutinates with concanavalin A, even with 10 micrograms per ml, while those isolated from asymptomatic carriers were unable to do so. And this first observation was published in Nature in Biology, and it started a whole new era in amoebic research, which I'm going to summarize here. So, since that observation, several other observations came about, for example, from Pete Sargent in 1978, where he started to describe different cytokines from isolates coming from amoebas, originating from patients with symptoms and distinguishing it from other patients there. Here again, another article by Pete Sargent. Then other groups started joining. John Akers, William Spice, we have Edward Tanny from the Bernard Knox Institute. We have again a group from in Japan that also started joining in. Groups from Israel, and again from the German group, from Mexico obviously, and another one from Mexico. And then the big question was, okay, is there one amoeba that is able to convert from a non-pathogenic to a pathogenic form? Is it really that first amoeba that is that one amoeba that is able to do so? Well, in 1993, these two scientists, Dr. Louis Diamond and Graham Tarr, were able to do the first academic culture of that, of an isolate coming from an asymptomatic person that was infected with the amoeba. And this marked a whole new era, although not immediately, because it had to wait a little bit more until something could be done. So until 1997, there was still the doubt, is it one or other two parasites? So in 1997, there was a big conference, a seminar on amoebiasis, and Valerie remembers our meetings on amoebiasis. Now this was in the framework of this seminar that took place in El Internacional with the support of the Pan-American Health Organization. Many scientists from all over the world to discuss this issue that had been increasingly pressing the advancement of the subject of whether there was one or there were two amoebas. So meeting, it was concluded that there were really two different species of amoeba, and it was supported and published by the World Health Organization, in which citing this meeting in Mexico with all these researchers, it was admitted there are two different amoebas. It is nice to remember when we were a little bit younger. So all researchers participated in the meeting, but I am particularly proud to see Adolfo and myself over here some years ago, not so many. But after this meeting, immediately there came an explosion of research done with both parasites and having the arsenic cultures of the Camiba dispar that were made by Dr. Diamond and Clark. There was another big initiative in the field of amylase. Namely, it was the Camiba Genome Project that was held the year after, in 1998, in October this time, in Rockville, Maryland, at NIH. And we can see here Dr. Diamond, we can see Dr. Adolfo Martinez Palomo, here is Exorganic, and here is Graham Clark, just to name a few. Now, at that time, I was working at NIH, and and at NIH, after spending 2 years, I was returning to Mexico City in 1995, and I asked Dr. Diamond, would you please give me a sample of your dispar cultures so that I can take them to Mexico? And he was a very generous person, and he said, oh yes, Marta, I will be very happy to share it. And I want to have more work done in Mexico City. Now the problem was that this Camiba dispar, the arsenic culture, was very difficult to culture. It was really very few hands could continue to do that. So we had to plan a carefully designed strategy for exploitation of this parasite. And so what I did is I bought some socks. I went to the lab with Dr. Diamond. We made sure they were very nice cultures. And I took them and trained in my socks to come to Mexico. And those are the only arsenic cultures that were available, that have been available in Mexico since. There was no other way to get these arsenic cultures. Why? Because the amoeba did not like the cold, did not like the heat, and did not like to be frozen. So years after, we were able to develop a method we could freeze them and maintain in the lab. So just to see how this impacted, we can see how many publications from Camiba histolytica were started since 1945, and you can see the surge of publication of scientific publications since having the culture and the meeting recognizing the two parasites as two distinct entities. And when I arrived back in Mexico, we started repeating some of the original reports from Adolfo. So we have here, when the amoebas are put at 4 degrees, they are separated. But once we added concanavalin A, the Camiba dispar agglutinated, but the Camiba histolytica clearly made a much bigger agglutination. We started comparing also in the light microscope, and we saw that the original drawings that Emil wrote made in his publication in 1925 really reproduced what we were looking in our microscope. And the same thing, we could see some differences where the much smoother surface membrane of the Entamoeba was in contrast to the rough surface of Entamoeba histolytica. We saw the movements and the specialized attachment organs of the two amoebas. We can see these forming big moving structures, but small phylopodia, for example, while Entamoeba histolytica had all these vacuoles on the surface, on the rough surface, and also was attached very strongly to the surface. As described before, Entamoeba histolytica has a very large surface projection of the sugars that explain the agglutination, the much better agglutination compared to Entamoeba dispar. And also, we could see the cytopathic effect on cultured monolayers. We can see that Entamoeba dispar was much milder on the poor cultures than was Entamoeba histolytica that destroyed the cultures very, very well. One phenomenon that had been described before was redirection of surface antigen, for example, in the presence of antibodies. And this is a phenomenon called capping. Well, capping had been described and is very prominent in Entamoeba histolytica when put in the presence of patient serum, but not so in Entamoeba dispar. And when we looked at it through electron microscope, the difference was also very obvious. We can see these large projections, which we are not sure, but we think that the amoeba might take it as a way of releasing antigens into the medium, so that they distract the attention of the immune system, while they happily go somewhere else in the tissue. This is not seen in Entamoeba dispar, or is seen in a much lesser way. Erythrocytosis, it is strikingly the difference between the two. We could identify the hemoglobin content. Here is Entamoeba dispar. It is at the same time 5, 10, 15 minutes of putting them together with erythrocytes, and we can see the difference that is here in Entamoeba histolytica. Again, in the surface, in the place where the amoebas attach to the substrate, we can see that histolytica even forms filaments that probably Van der Leyen in electron EM will be able to describe much better, and they are absent in Entamoeba dispar. Maybe we can see some filaments over here, but they are not exactly in the place of the parasite attaching to the substrate. And also, when we covered the slide with fibronectin, we could see that the actin, that polymerized actin identified with laminated phalloidin, was concentrated exactly in the point of contact with the fibronectin, but not fibronectin. So our in the liver, in an animal body. And in hamster, what we could see is that after injecting Entamoeba dispar directly into the liver of the hamster, we could see amoebas very surrounded by a lot of immune cells. Later on, we could see really a big concentration of immune cells, and at 25 24 hours, even this concentration was receding and regeneration had started. When we compared what happened after 6 days with Enterprise, there is hardly something to see. Everything is repaired, while histolytica is increasing and it's really damaging the tissue. So finally, when we tried the intestinal model, we added this part, and after long periods of time, these trophozoites were unable to penetrate. So we have some today, and that is that amoebiasis is still a health problem in many parts of the world. And in these parts there is no access or little access to potable water, and this is not widely available. The recognition of the existence of these two species was a major breakthrough in the field, and it involved the participation of many researchers around the world. The seminars that gave place to this meeting were periodically organized in Mexico City, and they really provided a fertile ground for collaborative discussions. Acceleration of this part resulted in a tremendous increase in research in the field, and we could do a lot of comparative studies in this organism. The sequencing of the genome, first in histolytica, and then in this part, allowed great advances on the mechanism of pathogenicity of this elusive metabolite histolytica. And finally, after 100 years since the first observation of immunobrood on the existence of these two species, there is still much to be learned of these two parasites. And so, just to mention some of the work was done by Daniel Lamas, by Anel, Liz, Carla is here, Viviana, but Adolfo, I want to show you a better picture. He says hello to everybody. He would like to be in this presentation, so I brought him. This is the group of my lab nowadays. And we have also moved to another subject, which is the organoid development. And this is why I wanted to show this. It was a symposium that we organized, and only to introduce you to our general director and to our academic secretary. You have met Carla and Monica. And science forges lasting friendships, like the one I have with Iris, who is our next speaker. Thank you very much.
Thank you very much, Marla, for this great overview about 100 years of Indonesia. So, are there any questions? Marta, nice coming to the history of these two amigos. I have a question. Now that you show in the animal model that it was time taking the difference between this part and histolytica, one is resolved and the other goes to the abscess. Can you check in the in vitro assays if it's also time dependent, that this part will show more agglutination with glucosamine or more digestion of the fibronectin, like you showed? Yeah, no, we have extended the time of incubation with fibronectin, and there is no digestion. We have also extended the time of everything, basically, of the amoebas die first before they are able to eat. And even when you inoculate directly either into the portal vein of the animal or in the liver, the immune system will take care of it. So, and
models with colonic, trying to see if there was invasion in the colon. They failed to show any invasion. Now we can do genomes already. Anybody found the major differences among the two strains? There are not major differences, but there are differences, and this is our next topic.
You mentioned that in Congreso is a non-pathogenic amoeba, but this is all the cases all the time. For example, little change in immunocompromised people or even in a lockdown mouse.
When it was first described as non-pathogenic, really what we describe now is that it is non-invasive. It is able to cause some very superficial damage, even in immunocompromised people, but it does not invade.
Thank you.
I have two questions. With these match and phenomenon. I just finished writing a review of my cousins together of microbiology animation. And that was the first description of what we now call extracellular organisms. I don't know if you are members of the group characterize better these vesicles in terms of the content of binding, RNA, or tetracycline, or something. Not all it's just the first. The second is about the microbiome of animals. In fact, I think you use the chemistry, right? Yeah. Effectively, each part of this you have the same microbiome.
Well, I answered the first question first. That was part of my doctoral thesis. So it was very difficult because the protein pattern depended on what you use to induce the capping. If you use concatenated A, if there was one specific protein pattern. If you used patient serum, it was another pattern completely different. If you used surface antibodies, that was another pattern. It is very difficult to know which are the proteins that are going to be redirected into the capping and then released into the medium. At that time, which was very recently, we did not have as many tools to analyze these vesicles, but we certainly have the tools now. We should do it. And the second question was the microbiome. Yes, we did not do it at that time. And right now we're having problems in cultivating the axenic amoeba dispar. I actually called Graham back to ask him, please help me. I might not put them in my socks again because I will not be able, but somehow we need to bring it back here. But that would be a very good thing that can be done without problems now.
There is a curiosity. The isolation of the spine and the story but there are some reports that you can find two both in one patient.
Yes, you can find both infection with both species in one patient. What happens there? Mostly patients suffer from diarrhea, malaise, because of the use of metronidazole in other infections, the invasion into the liver is less and less frequent. So it is difficult to make an extrapolation of whether having both amoebas, for example, will bring forward a bigger possibility to have invasion or not. So it is hard, but it is possible to have both.
Okay, if there are no further questions, again, thank you very much, Martha. Thank you.
So it is also my pleasure now to welcome Dr. Iris Bruchhaus. Dr. Bruchhaus, she graduated from biology in Hamburg, Germany, and obtained her PhD in zoology. She continued her doctoral education at the Bernard Nocht Institute for Tropical Medicine, where she is now working as a full-time professor and leads a group on host-parasite interaction. She has been focusing on the cytoaddition of the malarial parasite Plasmodium falciparum with the blood vessel endothelium of its human host, and on the tissue invasion process of the parasitic protozoa Amoeba histolytica, which we now describe. Since 2021, she is part of the board of directors of the Bernard Nocht Institute for Tropical Medicine, and she is reviewing board member of the Deutsche Forschungsgemeinschaft, the agency, the funding agency, one of the funding agencies of Germany. Furthermore, she is part of the board of several scientific societies, and she is a great researcher and a friend. Welcome.
Much, Martha. So I'd like to talk today about our studies on the invasion process of Celular. And I'd like to start for those of you that are not familiar with this parasite, very short with the life cycle. So the infection starts by taking up cysts with contaminated water. The cysts go through the stomach and the intestine, and then in the colon the exudation takes place and the parasite by division. And at one distinct point, the parasites form the cyst again, and the cyst will excrete. So far, so good, nothing happens. And what can happen, and already Martha mentioned, the amoeba can get invasive. That means that they disrupt the tissue mucosa and can induce amoebicolitis, or they go through the mucosa into the bloodstream and form liver abscesses. We have only odd numbers of an infection rate of Amoeba histolytica. There's one from 2016. It's postulated that we have about 100 million cases of Amoeba infection, but only 26,000 deaths in the year 2016. Therefore, only a minority of infections really due to invasive disease. Here are some of the fast movies, some of the Amoeba in the culture, and the name Amoeba histolytica comes from its rupture of tissue, and this is shown here. So we have a monolayer of fibroblasts with pure amoeba sitting on this monolayer, and within minutes this monolayer is totally disrupted by the amoeba. And in this context, I'd like to remind you, as I mentioned before, that this will be important for our study, that Amoeba histolytica proliferates in its colon. So our first research question is how do Amoeba histolytica interact with intestinal cells, and this work was done by my former PhD student and now postdoctoral in Anlass, as well as a PhD student in Biología and our master student in Rebecca Allen. So we're working with two Amoeba histolytica clones, both derived from the classical isolate
that are not cleared within 7 days post-infection. And where do these clones come from? We have originally two Entamoeba, we got two Entamoeba strains from the American typewriter collection. One we turned A and the other one we turned B, and we used them to infect animals. And it turns out that A did not was more as non-pathogenic, whereas B was pathogenic. Then we cloned these clone strains, 12 clones of A1 and 12 clones of B2. And what comes out is that all 12 clones of A1 are non-pathogenic, whereas the majority of the B clones, some of them are totally non-pathogenic and others are highly pathogenic. So at this time we decided to do all our studies with the clone A1 of the non-pathogenic one and with the clone B2 of the pathogenic one. We did a lot of transcriptome studies and there are only a few genes are differentiated expressed, about 70 genes are differentiated expressed between those proteins. The problem to study this invasion process is that there are only a limited amount of animal models present and the ethics for these models, so it's hard to get the ethics of these sometimes strange models. So we started to work with the tested organism like Celular. And for this we isolated clips of the small intestine and the colon of human patients. The clips were isolated, they were put in culture, a 3D culture where we get these so-called steroids. These steroids were then disrupted to single cells, and the single cells were sealed into a monolayer and differentiated into an intestinal epithelium, which is shown here for a really nice monolayer. This work was done in collaboration with a colleague of the Congreso Institute in Berlin. So how is our experimental design? We add our pathogenic and non-pathogenic Entamoeba to the monolayer, and then we measure the tier, which is the transepithelial electrical resistance. That is a quantity to measure the disruption of the cell layer caused by Entamoeba. Then we perform microscopy, visualize the invasion process, and we also perform transcriptome analysis to analyze the change of expression of the human intestinal cell as well as so first results I'd like to present you are the interaction with the small intestine. I'd like to remember that normally Entamoeba is not present in the small intestine. And what we see here is that when we use 500 what we see here is that there is over time no obstruction of this intestine. And this is also what we observe here in the microscope, where we have two markers for these tight junctions, and the conjugation with these two are the exactly the same light structure. Therefore, we can say that A1 triplosides is 500 are unable to destroy the epithelium of the small intestine. Now coming to the colon, this is now observed. So here again, we add 501 and 502 triplosides to this monolayer. What we see here is that the monolayer is much more fast and disrupted by adding these two triplosides. And after 24 hours, we see also disruption of the Entamoeba, and this is shown here. This is the picture after 24 hours, and this is really total destruction of this tight junction. Sometimes there's this big hole into the monolayer. We cannot see the Entamoeba and they are washed out by this fixation. So what we say here is that 502 triplosides can destroy the colon epithelium more effectively than 501 triplosides. And then we start looking at the transcriptome, and because we also want to analyze the transcriptome of Entamoeba, we have to add more Entamoeba to this incubation process. In this case, 10,000 A1 and 10,000 A2. And now these Entamoeba disrupt the intestinal monolayer, however, again, no difference between A1 and A2. And when we look at the genes that are up and down related, what we see first is that at the beginning of
Then we look at what are the pathways that are regulated, and it comes out that the response is similar, either if you have A1 or 2 amoeba, in all cases, mainly pathways involved in the biological pathways are slightly accepted pathways, development sector, and other pathways, and it's the same for everyone. Again, I have to mention that the response is same when you have a 2 amoeba, then you don't see any reduction of the So what we can say is that A1 and B2 results mainly trigger accumulation of genes in cells as a small percent involved in the immune response. And how does it look now in the world where molecular live? Here again, if you use 10,000 molecular, you see a normal destruction after 4 hours adding 10,002 in comparison to 10,001, so that is very similar to what we saw with 500 molecular. And when we look at the transcriptome or the amount of genes that are down-regulated, again what we see is after 4 hours that more genes are up and down-regulated in response to the one. This is shown here, these pink colors, and much more genes are up-regulated in response to the one after 4 hours than in response to the two. However, exactly the other way around, after 8 hours, here much more genes are upregulated in the 2 after 8 hours in response in case in comparison to A1. And this comes more clear here, only about 20 genes are upregulated after 8 hours, and more than 100 genes are upregulated in response to 2. But nevertheless, the total difference is more than 1,000 genes in this time. So common cell response to cells starts later, but is more stronger than the response to the one cells. And this was also shown here in the heat map. What you can see here is the top 30 differentially expressed genes, and most of these top 30 belong to the group of 8 hours co-incubation with the 2. And when we look in detail, and all these are marked as errors, what kind of genes are these are mainly genes involved in pro-inflammatory and antimicrobial chemokines, phospho- and NF-kappa B pathway, as well as T-cell regulation. Now coming on to the regulated pathways. So as I already mentioned, on the few genes that we have more or less no significant pathway in response to the 2 amoeba, but in response to the 1 amoeba after 4 hours, we saw that these genes are mainly and after 8 hours, we have here no identified pathway because these are only 20 genes or 19 genes. And when we look at the 2, again, we saw this increase of pathways involved in the initial response, like TNF, development, and development, and so on. So co-incubation between 1 and 2 cells results in a overlap, as shown here. So the response is not the same, it's very clear overlap. And into 2 cells trigger upregulation mainly of genes involved in the regulations. And now the last question is, did the amoeba respond somehow to co-incubation with the monolayer? And then we'll look here for co-incubation with the colon cells. We saw several hundred again, 300, around 300 response in response to the colon cells. However, the problem is here that the activation of the amoeba gene is very bad. So approximately 50% of the genes we identified including 5% of the proteins. However, when we look in detail, we saw again here in the 2 amoeba that most of the genes that were annotated and that were upregulated are somehow involved in stress response, optic stress response, but also in some proteins that enable proteins. So this is how I can come to summary of the first part of my talk. Concerning the small intestine, we saw that A1 and B2 trophocytes are unable to destroy the epithelium of the small intestine. However, there are several hundred genes that are upregulated vascular accumulation of genes mainly involved in the pro-inflammatory and anti-mitotic chemokines like phospholipid plasma and T-cell regulation. In case of endometriosis, what we saw is a fast accumulation of genes including antioxidant proteins and DNA proteins in between tryptophanes compared to the one. And with this, I'd like to come to our third research question, and this was mainly done by Barbara Monica. She's now a postdoc in our institute in the group of my colleague Hannah Locker. And she asked the question, what is the function of the extracellular diseases secreted by interliga? So interliga secrete like most organisms, most cells, secrete extracellular vesicles, but so far we have no idea what these vesicles are for. So again, here our experimental design, in this case, we work with the one and two tryptophans. We calibrated them in these 6-rod plates, collected the supernated after cultivation, and isolated the vesicles by differential centrifugation as well as alpha centrifugation. Again, with very small time, and so we had these isolations from A1 as well as and what we did next, characterize these, then we performed to determine the cargo of the vesicles as well as the microRNA transcription of the vesicles. I would like to skip this point today. And at the end, we asked ourselves what maybe is the function. So we used monocytes isolated from male and female mice and tried to see if these monocytes can be stimulated by the vesicles, and use male and female mice because in case of liver upset, there is known that there is a sexual dimorphism takes place. And we analyzed these stimulation by flow cytometry, transcription analysis, multi-site, and this is the analysis response of monocytes and stimulation. So first coming to the characterization, we first looked at the particle size of A1 and tryptophan, that's a bit different. Then also the amount of particles or least secreted, that's also different. And then we looked at these vesicles really from an amoeba, secreted by an amoeba. So we stain them with the surface protein of the classical surface protein of an antibody against lactactin, and we see there's nice particles or protein is present, and also we use an antibody against lipophosphatide, and again you see here staining of these vesicles. So these vesicles are really from Analytica. In summary, there's no difference in size and number of secreted LEDs, and we were able to identify surface molecules on the surface of the. So next we look at the proteome, so the carbon with the proteome of the tryptophan and one of the tryptophan. We identified approximately 2,500 to 2,700 proteins, and also the carbon of the release of the release are more than 28,800. Here we have the problem of the low amount of hypothetical protein, approximately 40%. But what we can say is that we've got an enrichment of proteins that contain transmembrane domains, so from the total 30% to 30%, and the proteome, as well as the same thing is true for the signal peptides. Here we got an enrichment. Look at the molecular function of these proteins, and we saw an depletion of proteins somehow involved in nucleus function, so for example DNA binding, but we saw an enrichment of proteins that have typically bind activity, activity of pyrophosphate activity. And what was also obvious that when we looked here, there are differentially expressed proteins that we A1 and A2, and these are differentially expressed proteins in tryptophan, but we saw not only an overlap of 50 proteins, that is the protein of the yeast. And now most interesting is found a lot of molecules that we know are the host environments of Amoeba, for example, the systemic activities. So, the enrichment of proteins and what environments, secondly was found, these contain viral factors and antioxidants, but unfortunately, especially for the differentially expressed proteins, most of them are hypocytic. So coming now to the stimulation of the monocytes, so is there any function of this? And here first I have to introduce you to sort of monocytes, the lysis high and lysis low monocytes. The lysis high are pro-inflammatory and I have pro-inflammatory and antimicrobial function, and the lysis low are controlling monocytes that go into the vascular system and they are more responsible for activation tissue. And as a marker for activation in this area, we use CD38. And the factor analysis is shown here. So we use mock controls, and what you see is in comparison to mock controls, we got an increase in the amount of activation, monocyte activation, either in the 1 degrees, but also in 2 degrees. But when we heat inactivated CDB, so what we take is an activation, but also is coming from the tissue activation. And this is also the same using our lysis low monocytes, and inactive activation is not as high as for lysis high monocytes, but again, we saw an increase in activation after adding increase with the 1 as well as inactivated, the 1 is the stimulated monocytes inactivated. And then we so is there any difference between 1 and 2 of the transcriptome after stimulation of the monocytes? There is no difference. There is no difference. There are mainly chemokines are activated. So a 1 stimulation mainly regulates the nuclear function for the cytokine response, and this is the same for cytokine secretion after stimulation. Here we look at cytokines that are secreted, and we see no difference. However, again, after inactivation, we don't see any secretion of the cytokines. So in summary, there's no difference that one and two increase the size and number of CDs. These metabolites can be detected on the surface of CDs. We saw enrichment of proteins involved in biogenesis and signaling. These contain viral factors and antioxidants, and unfortunately, most of the different virtual so these can stimulate monocytes, and this stimulation may activate molecular function involved in cytokine response. And you see that after activation, each disease cannot have any. And with this, I'd like to thank the whole group, so far of people working on the Amoeba, and I'd like to mention again Julian Anna and Barbara Omanika, so one part working on the Amoeba and the other part working on the Plasmodium. All our collaborators, all our fundings, and I'd like to thank you all for your attention. Thank you very much.
Thank you very much, Iris. And I would like to ask if there are any questions for the talk.
We look for it, it's almost part is skipped. So there are microRNAs in the vesicles, a lot of microRNAs, and the problem is when we look what genes are targeted, so maybe the whole human genome is targeted and maybe the whole animal genome is targeted. So at the moment, and however, we only find one differential microRNA that we want to mention, but several hundred microRNAs.
I have a question. At one point you said that many pro-inflammatory genes are
respond to the presence of cellular. And I think that is exactly what you see also in the cellular abscess. There's a fire in the immune system, and the cellular are survived in the abscess, and they are in the border of the abscess, and all the other parts of the abscess.
But in your own words, you didn't have an immune response.
But you're right. But the epithelial cells, they can also respond. So, but you're right, but that is the next step. What we like to do now, the next step is also in these models, we have no glucosa, and we have no microbiota, and that is the next step we like to do, we have glucose and microbiota, also in the cells.
And have you thought about using human microbiota or infected people?
Yeah, but it's not bad. Yeah, that's right. It's not when it's such a model, it's kind of
Okay. Thank you very much. Thank you very, very much. And I would like to invite Dr. Rosanna Arroyo. She is going to talk about Trichomonas vaginalis, another protozoa that infects the human. And she has a BS in chemical pharmacobiology from the University of Guadalajara and a PhD of molecular biology at Celular, which is the institution we both work for. We both and several other people from the audience is one of the, it's bad that I say it, it's one of the best research institutions in our country. And she has had a postdoctoral position in the Department of Microbiology at the University of Texas Health Science Center in San Antonio, Texas. And in 1992, she joined the Department of Infectomics and Molecular Pathogenesis at Celular, where she is currently a full-time professor. She is the coordinator and the head of our department, and she has collaborated with other researchers and published a peer-reviewed paper. 5 articles in PDX journals with over and more than 340 presentations at national and international congresses, invited more than 80 times as a guest speaker with 11 Mexican patent titles. She has mentored around 50 graduate students, 20 PhDs, 30 masters, and 7 students, and 7 of them are already independent researchers in other institutions in the country who are part of the national system of researchers. Her contribution to the field of amoebiasis was the identification and localization of antihistamine, of Entamedes politica, and but she is now moving to another field of expertise, which is trying to identify, localize, and characterize the pathogenic mechanisms of Trichomonas vaginalis. Her studies on the identification and characterization of the virulence factors of these parasites that are expressed during infection and influence in glucose and iron on gene expression have led to the identification of 2 mechanisms of post-transcriptional regulation by iron in Trichomonas. And her data led to the development of 11 patents, as I said before, that include the development also of a diagnostic kit for trichomoniasis. She has received several national and international awards and recognitions like the Nolan Officer Award, California Prize 1999 in Basic Science, the first place in the 17th National Research Award from the GlaxoSmithKline Foundation Award in 2006, and the UNESCO Equatorial Guinea International Prize in 2012 for her contributions in life science. She is now Researcher Emeritus at the National Research Institute and has been awarded the 2023 L'Oreal UNESCO AMC Funales Prize for Women in Science. Thank you very much, Rosanna, and I would like to thank the organizers for this invitation and have the chance to show part of the work that we have been doing with the groups that have been in my lab. In this time, I will talk about the peptidases as part of the virulence factors that Trichomonas has. In particularly, I will talk about two cysteine proteinases called legumines that are part of the cytoadherence and cytotoxicity events that Trichomonas does to survive in the host for a long time because it causes a chronic disease. I will talk about a little bit of the disease, trichomoniasis, the parasite that causes Trichomonas vaginalis, and part of the pathogenesis to remain in the host. The peptidases involving virulence, very light, and especially the legumines, 1 and later. Trichomoniasis is one of the sexually transmitted diseases that is the most common non-viral STI, and it can reach about 180 to 270 million cases worldwide every year. So it's a very common disease worldwide. This disease is, because of that, a health problem, and is considered a neglected disease because not many people is involved in this, because it's one of the diseases that affect women mainly. It also affects men, but the most that suffer this disease is women. So there is treatment. However, the problem is that most of the infected people are asymptomatic. About 85 to 90% the people infected don't know. So we need a better system to give the right treatment. Trichomonas, this disease, those people that are symptomatic will show prostatitis and vaginitis, but even though it's a chronic disease, it is a risk factor for an HIV infection and also for cancer, prostatic cancer, and cervical cancer. So if you treat Trichomonas, you can reduce the appearance of cancer. It also can reduce at about 20% the HIV infection. So we need to know people that is infected and receive treatment and reduce these serious health problems. These Trichomonas can also affect pregnant women, and it causes a serious outcome. There is premature labor, low weight of the newborn, and the infants can also be associated with infections such as respiratory or vaginal. It appears to be very simple. The cell cycle by binary fission. Apparently, there is no sexual division, even though there are some genes that appear to be part of the meiosis, but there is no demonstration yet. This is the parasite, how it works. These parasites are different to other protozoa. Doesn't have several stages, but it has several morphologies. This is the typical one by scanning electron microscopy. It's the molding one, but when the parasite is attached to vaginal epithelial cells, transform completely to an amoeboid form to be more adhesive to the epithelium, avoiding the big flush away during the menstruation or by the vaginal secretion. and also can present a resistant form. The system-like morphology grounded. This was published by Benjamin some time ago, where we can see the parasite internalize the flagella that are here, and also the actual site, and convert in a round form that is resistant to nutrient deprivation, to cold, is a resistant form. Let's see about in the beginning that this was but now that is also active and can return in the appropriate position. So this is, if not compared with those micrographies that already Dr. Manuel showed about Trichomonas, but it helped me to introduce how it organized in the interior. It has a very complex cytoskeleton, as he showed, a single nucleus, multiple vacuolas, and also a single organelle that is a specific organelle that is called hydrogenosome that uses instead of mitochondria. This is one of the main characteristics of this parasite. And it also has a very structured cytoskeleton Golgi apparatus. By using different antibodies specific for actin or for tubulin, we can see by confocal microscopy how these structures are coordinated up all the way to the interior of the parasite to the axial side, and also the nucleus. The yellow, the green one is actin, and the red one is tubulin. Parasites and under division, there is some rearrangement that cytoskeleton undergoes before the two nuclei are separated. And also, we can see by specific antibodies how the different hydrogenosomes are localized in the parasite, or even by specific antibodies the Golgi apparatus. Confocal microscopy can help us to really appreciate the different organelles in the parasite. In the pathogenesis of Trichomonas, this parasite is really responding to different environmental factors such as temperature, glucose, pH, polyamines, zinc in men or iron in women, and also the microbiota. All of these factors are affecting, and the parasite is responding by controlling different properties in virulence that are important, such as growth, multiplication, aggregation, nutrients acquisition, site adherence to different epithelium of the morphological transformation that I showed you in the beginning, but also cause damage as amoeba does in some of the epithelia in the vaginal or urethra region. It can induce apoptosis even to the epithelial or the immune cells, but also can cause damage to the erythrocytes, can phagocytize different components and different cells that are present in the vaginal environment, and it has a very strong response to avoid the immune system once it's there. We are involved in characterize and study peptidases as virulence factors and how these are modulated by iron and glucose. A postdoctoral in my lab and PhD students, Cero and Rodolfo, help us to identify the beta-goma that the parasite has, the active one, because Trichomonas is very complex, and in the genome we can find 440 genes encoding different proteases, but trichomonas, by checking those proteases that are active, we can find about 30 different proteins. So where are all of those genes expressed? We don't know yet. Spectrometry, we have identified several of them, and some of them has proteolytic activity if we use seismography in 1 or 2 dimensions, and we can see how proteolytic activity bands are, or we a single proteolytic activity in one of the bands. So we have studied several of these, and I have named some of them, and I concentrate in this cartoon the different aspects we have studied with the system protein under different environmental iron conditions. Iron restriction conditions, the parasites express several of the system proteases, 39 and 2. They are involved in cytotoxicity and apoptosis induction. If we check high iron conditions, the parasite is also responding to several molecules overexpressing, such as PFO that go to the surface and participate in appearance, but also TRLM1 that goes to the surface and, as you will see, participate in cytoadherence. And there are others, such as CD4, that participate in hemolysis. So, how we end up demonstrating that some proteases participate in adherence? Well, we use a model, trichomonas, in the presence of vaginal epithelial cells or HeLa cells. We check the attachment and then we compare how this is occurring. The first papers help us to demonstrate that the proteolytic activity on the surface is necessary for adherence. Later on, we check by doing a ligand assay, fixed HeLa and proteolytic extracts from trichomonas, and we detect 2 surface proteinases that bound to the surface of HeLa cells. And these help us to identify one of the proteases, CP30, which is involved in adherence. How we demonstrate it? We produce antibodies against this protease, and using the pre-incubation of parasites with trichomonas before interaction with HeLa at different concentrations, and we found that this antibody reduced up to 50% the levels of cytoadherence. So, how complex is this region of 30 kilodaltons? Josefina, in his PhD studies, she demonstrated by seismography 2-dimensional that under different iron conditions, low iron and high iron, there are more proteolytic activity in the 30 kilodalton region than in the high iron conditions. So the question is, which of these proteases are participating in adherence? So she started for sequencing the N-terminal of this protease, and with that we found a gene, and this gene was cloned by Francisco in his MS and PhD students. He produced the recombinant protein, purified, made antibodies, and with these antibodies by immunofluorescence, we found that this protease, TRLM1, was on the surface of the parasite. Using these antibodies, by re-incubating the parasite with the antibodies, we block this protease, and we find that by interacting with HeLa cells, the protease activity was reduced, and the cytoadherence was also reduced. So this helps us to demonstrate that TRLM1 is one of the proteases that are necessary for trichomonal adherence to epithelial cells. We also plan to start analyzing another environmental factors such as glucose and see how this affects the trichomonas virulence. What we found was, first, that glucose is important and increase the growth of the parasite as compared with parasites that are under glucose restriction. Also, glucose restriction alters the cytotoxicity levels of the parasite. The parasite is more cytotoxic under glucose restriction than under high glucose condition. And also affect the amount of the different proteolytic activity, amounts of protein and proteases. For example, tyrosine is regulated in glucose, but most of the proteolytic cysteine proteases of trichomonas are down-regulated by glucose, are more expressed in high glucose. And this, by checking in seismography, we observe there are more proteolytic activity in restriction than in high glucose conditions. And this, if we check one of the proteases that are upregulated in glucose under seismography to be using hemoglobin, we can see that this protease is also overexpressed in activity. So now we can see that trichomonas can respond even to iron as to glucose, that we can concentrate in this cartoon. There are some negative response to glucose by overexpressing proteases and causing an increase in cytotoxicity or apoptosis, but there are also responses to high glucose by mobilizing to the surface and even to secrete of them. So this parasite is able to respond to different environmental conditions. So trichomonas, by checking the sera from patients with trichomoniosis by 2D immunoblood, we found that immunoproteomics, and we identified that some of these proteases are immunogenic, and some of them are important in our study, TEV11 and TEV11, and others proteases. This is a normal person serum that no reactivity is found. What are the legumines? This is the 3D structure of a legumine that is complex. And the legumines are peptidases that are also called vacuolar processing enzymes or asparagine peptidases that in parasites they are only described in Trichomonas vaginalis and Trichomonas vaginalis. These proteases are made or synthesized as precursors that need to be activated to remove those parts of the molecule that are interfering with the proteolytic activity. And this process of activation is depending on pH. But also these proteases are inhibited by different ways. The same components that are blocking the active site or by different components that we can add for control this activity. In trichomonas, there are several, as I mentioned, different genes, and they form different families that belong to different clans. The legumines belong to the clan CD and family C13. And these in Trichomonas, according to the genome, there are 10 different genes that are part of this family. And some of these are apparently not expressed. And those that I showed you that are immunogenic, that are LEG1 and LEG2. How are these proteases? How similar are they? Can we differentiate by using a specific? Can we detect them in the parasite? Where are they located? So we produce the protein, the recombinant protein, purified and made antibodies. And these are the two-dimensional Western blot that show that antibodies against LEG1 is detecting the proteases that correspond to LEG1, and antibodies against LEG2 are recognizing the one that corresponds to LEG2. And this is how we made it by mass spectrometry. With these antibodies that are specific to each one, we localize LEG1 on the surface, that's the yellow signal that you show here. By electron microscopy and we can identify in some vesicles, and some of these vesicles correspond to lysosomes, as you can see here in yellow, and some other vesicles in green that are not localizing with the lysosome. We can also identify this protein in hydrogenosomes, and by using Mito-Cracker, we can detect this signal. We can find also this protein in the apparatus. This is one of the collaborative works that we performed with Marlene Benchimol. And with this antibody, we can also detect this horseshoe kind of image that represents the Golgi apparatus in the community. So, as I mentioned, TBLEG1 participates in appearance by using specific inhibitors for legumines. We reduce enormously the amount of other proteases that when we use a specific for catepsinel proteases, we also see a reduction, but the major reduction is obtained with legumine specific. And the antibodies against the legumine, LEG1, we can reduce up to 60% the cycle appearance. So what happened with LEG2? LEG2, we can see it also on the parasite surface, the yellow signal. We can see it in vesicles, even on the surface, but also in the inside of the parasite. Some of these proteins are in the hydrogenosomes, that is confirmed by using the PFO as a marker for hydrogenosomes. We see a lot of yellow signal. And we also can see it in autophagic vesicles, that some of them can be detected when we use another marker for autophagy, such as ATGA, that is this yellow signal. But we can also detect it in vesicles that contain material that correspond in yellow. Some of these signals correspond with lysotracker. So what happens? They are appearing in the same organelles. Are they working in the same virulence properties? So if we do co-localization assays using both antibodies following both proteases in the same parasite, what we found is, yes, they are there, but they colocalize little, especially under high glucose condition, they polarize to different sides of the parasite. Lego 1 is close to the flagella, is close to the action side. So what they are doing, why are they at the same time, but why are not colocalized? So this is a zoom of these images. And if Lego 1 is working, I don't show data about the regulation in glucose, but if Lego 1 is in high glucose condition, we did a kinetic following by fluorescence how the destruction of the HeLa cell monolayers occur at 15, 30, and 60 minutes on their. See that the monolayer, by staying to, we see how this monolayer is being affected. If we follow the LEC2, we found that this protein in red is inside the parasite, is on the surface, but also is inside of the host cell. So HeLa cell is being affected by the parasite by using one of its molecules, such as TBL2. This is work in progress. We are working to demonstrate that in is working in cytotoxicity, whereas TBL1 is working in cytotoxicity. And this is our take-home message that trichomonas use two legomains that are localized in the same organelle in opposite parasite regions. They are both virulence factors that play different roles in adherence and probably in cytotoxicity. And they are differentially regulated by iron. So thank you very much for your attention, and I would like to thank all the students that have been in my laboratory, the different support, financial support I have received, the hospital that helped us to obtain human samples to do all our studies with the immunoproteoma, and also detect some of the proteases or some of the molecules we study in the vaginal secretions or antibodies against these molecules on the serum. And several of the collaborators that have been helping us. And these are different institutions. And my family. Thank you very much for being here.
Thank you very much, Dr. Rosana Arroyo. Nice talk. And I would like to open the floor for questions. Yes.
I have one more general question because I want no answer. Like in endometrial anesthetica, also in trichomonas, there are a lot of proteinases, it's in hundreds. So, have you any idea why these two have so many proteases?
Well, we have the idea is that probably so many proteases help the parasite to adapt to multiple environmental conditions. So we have studied 2 or 3 environmental conditions, and when we check some of them, there are most of them the same, but they regulate or modulate differently. For example, TBL1 is upregulated by iron, but is downregulated by glucose. So the parasite is turning on and off different genes to adapt to
the different environmental conditions. That's the idea. And probably we don't see all of them because we don't know in which environmental conditions are they expressed.
Thank you very much. Are there any other questions? Yes.
Why do you use black pots and Mount Collins? Because in the region, the commoners prefer Mount Collins.
Yes, the standard medium contains maltose, but we switched to glucose because acquired glucose. It has from glycogen, it has amylases that can degrade and process the glucogen and introduce it to the parasite. And there are also another study from Petrus that also use glucose to check the transcriptional expression in the parasite, and he found major differences in the expression of some of the proteins. And some of them are the ones that we are studying.
Thank you very much. So, join me please with a big applause to Dr. Sanaro. And people know that I'm that is from Dr. Jaime Ortiz. He's a chemical engineer from the University of Guadalajara, a Master of Science in Bioengineering from CIMBESTAB, and a Doctor of Science in Biochemistry from the University of Texas Health Science Center at San Antonio. He joined the Department of Biotechnology and Bioengineering at CIMBESTAB in Zacatenco in 1995, and he is currently a full professor of this department, level 2 of the National System of Researchers, and he has been studying the protein biotechnology with emphasis on expression, purification, and refolding of recombinant proteins from protozoan parasites responsible for neglected diseases such as amoebiasis, trichomoniasis, and Chagas disease. His laboratory has expertise and infrastructure expressing, purifying, and refolding recombinant proteins using E. coli and Pichia bacteris expression platforms. Studies of proteins from protozoan parasites have contributed to the structure, function, characterization of these molecules, and are part of a multidisciplinary effort to develop new diagnostic tools and vaccines. Since 2011, he has been part of the multidisciplinary consortium formed by the Baylor College of Medicine, the Texas Children's Hospital Center for Vaccine Development, Tulane University, the Autonomous University of Yucatan, and CIMBESTAB for the development of the first therapeutic vaccine against Chagas disease. Thank you very much. You have the floor.
Okay, thank you very much. And first I want to thank you for this invitation. And talk about this journey from developing first therapeutic vaccines against Chagas disease. Well, this is a multidisciplinary effort, as Dr. Espinosa said, since 2011. Since 2011, we have been working in this effort. I want to show you these times how the journey has been, especially my biotechnology point of view, which is the achievements and challenges to explain and given to the test the clinical studies, as well as to develop for the gene technologies. So Chagas disease is one of the most important this slide that I Santiago one almost everything. But what I want to say is from more than 100 years with this physician, Brazilian physician Carlos Chagas, with the pioneering work of him, that still we don't have a efficiency cure for this disease. Well, here is the latest data from the WHO that this disease is 2021 is basically in America, but now with the change, there is also from the United States, Canada, and some countries in Africa. So there is probably this is a sub-estimated and 30,000 new cases in Europe and other new cases. There are 70 million at least, and there is causing deaths in Europe. So one of the cases in Mexico, that is estimated at more than 1 million, but there are some estimated that probably there will be about 4 million because the diagnostic is, there is no system to diagnose this disease. And also, most of the people that have infection is not in the rural, is in the rural areas where there is no available in the health system change. Most of them are really diagnosed when there is also the cardiomyopathies are present later stage. Well, the treatment. The basic treatment, but two of drugs that penicillin solely for 3 months. But there are efficacy only in the first weeks of infection, but most of the time are symptomatic and not effective. So in the chronic phase, there is no efficacy, and there are several secondary effects that a high percentage of the patient drop off the treatment, and that is why there is a need to therapeutic vaccines. So our unit started in 2000. We started working with the colleagues from Arizona and University of Arizona to develop a process to purify by chromatographic methods plasmid, supercoiled plasmid DNA, but to use as a DNA vaccines. So we start providing this plasmid to the group of Dr. Ayrton at that time in the Autonomous University of Yucatan to make a screening of antigens and selecting the best antigens for the development of this vaccine. So after many years, using the two models, animal models, mice and dogs, they found two antigens, PC24, which is a biological calcium protein, and TSF1, which is a surface antigen. So we started providing this, and the proof in those antigens were really the best. Well, in 2010, all of the results indicated that those antigens are really promising antigens to go for develop this innovative vaccine. But then together with the leadership of Dr. Peter Flores and Magdalena Watasa from the Massachusetts Center for Vaccine Development, with the support of the Carlos Slim Foundation, in 2011, there was an initiative to accelerate the development of this therapeutic vaccine against Chagas. The target profile that we propose is really a complementary therapy to bridge the tolerance durability of efficacy upon the therapy. But the goal is really delay or halt the progression of Chagas myopathy. In other words, this is the therapeutic vaccine, especially for those that are in the chronic indeterminate phases. So we start, well, let me say, at that time, there is no approval for DNA vaccine. So we need to think in another alternative. And the best alternative for that was using recombinant proteins. So we start using this, all of the infrastructure of all the institutions to produce those antigens. Well, Baylor and Texas Children's Hospital, for vaccine development, start producing the TC24, which is a small protein, with 24 kilodaltons. So the expression of this in E. coli was really very efficient. But when the protein was storage, they would start the application. As you know, one of the problems for this vaccine development, especially for protein, and the regulatory agencies like FDA or Congreso in Mexico, is that you have to have just demonstrate that there is no alteration in the storage. Because these alteration may be kind of studying so this protein has 4 systems, and fortunately, the mutation of those systems avoid the aggregation, and also the efficiency of the production was very increased. So we came to use until more than 4 grams per liter of that protein, and after the purification was more than 85%, and also was stable for weeks, and also good temperature more than 30 days. The process was really straightforward, that is suitable for the production. So, well, the PSA1 was alternative because only was producing bags, and because my lab was dedicated to the folding of protein. So we start working first in the production and the E. coli. Well, first we start with the very small yield, but after several events and optimization projects, we achieved to have high yield in the reproducible process that we achieved 3.5 grams per liter, almost the same as the TC24. But the problem is we have to refold purification and refolding induced combined product. Purification is not really the problem when you have induced bodies, but refolding is really difficult. After many tries and optimizations of double small issues steps, we found the process that was producing using this for step by one growing layers, one thing isolation. But you know, there is an advantage when you have nutrition bodies, because it's just in the isolation of nutrition bodies, you have 85% of the protein. However, to do the reforming of the molar mass, the contaminants, we have to develop a purification process that is not useful for the ancient protein, which reduces have to use ion change because they are allowed to use the tag for the proteins. And so we did the chromatographic reformat with this, but at the end we have purity greater than 94%, and you look small, but as you produce the protein was enough for the test also for the clinical and also for the production. But the problem here was the stability. So we checked And you see, measure beside an angular strategy, and as you see these two loads, one is just recently, and the other is 1 year, and it stores microceramic or even 30 weeks at room temperature. And as you see, basically there is no much difference. And also, this one of the things that we have in this consortium is we share the proteins and we test we develop the process developing something another from zero start. We reproduce or check the data from method on this children hospital, and then also check out with it. So what you say this is consistent. I don't show in my data. This is consistent with the simple start data. Well, this is the also the circular diagrams of both samples. And you see there is no difference, and basically both samples are the same. So we basically for the C21 now has been produced actually. The toxicity studies are completed and are ready for clinical phase 1. The consortium is just waiting for the approval of the president initiating the clinical phase 1 and Instituto Nacional de Nutrición, Salvador Subirán. While this is a challenge that we have is set up the TSA-1 for the year production. Well, here is just a cartoon. This is our journey that started in 2005, providing relief for the quality. We have been here, visiting 40 here, is waiting for clinical evaluation. The phase trial already accepted the municipal participant 1. Visited 4. Vaccine we have, GRA for child disease, when we start. Here is the prototype of the vaccine that we make. In the meantime, since the beginning, we start looking for alternatives in the group, also for new animals and new technology. Let's say, I can say that in this couple of problems that is suitable, we select Chacas. We select Chacas, which is responsible and very studied for a long time. This is an inhibitor from the major city from the cruise. And also, in that time, and the same group made a study to see if the PSA1 was really probably suitable for use the vaccine for all of the use. And here we found biologicals that are concept. So the idea is this vaccine is working all America. So with that idea, first using Sargassum substitute for the loop series, and these two loops, two reactors. And we use all available tools, bioinformatic tools. First, to obtain the model of the protein, and then we classify, instead of oxygen predicted solubility, we use we synthesize 9 of these 20 possible combinations, and then express it. And we found that this string expressed the chimeric proteins as much as. So, going furthermore, be able to substitute 4 editors, because my friend, epidemiology friend is well, not enough. We need to have more. Even we want to have 50 and 110. But putting one as a string, one another, has been proved that doesn't work because the protein goes to the inclusion bias and we come back again to the same problem. So two students come to my lab and to do the masters, and they decide, well, we will use and build chimeric proteins or with 4 antigens, with molecules. So there was about 120 chimeric proteins. They obtained, well, basically they're taking their pictures now, the AlphaFold platform. So they designed all the models, and they used the criteria of the AlphaFold just to discriminate many of those and select 2 of them. And one of these was incredible because we can express about 60% as a soluble fraction, and we can purify, well, it has been purified, and we obtain really very good changes. So, in thinking, we have at least 1 gram of soluble protein to be purified on this. Now, the text in an animal model is progressing in our facilities in Yucatan. And well, this is now this is the goal of the people that are working on this project. We start here, but now produce in Mexico this prototype of vaccines, and Mulliquid did a test of toxicology tests, and now I hope soon will be a start in the clinical phase, and also where the recommendation of the Johnson for this can be very especially the support of this. Well, this is my look at different terms, and all of them has been the ones that work. And Texas Center for Vaccine Development, the Centro de Investigaciones Regionales de Unamuchi, the National Autonomous University of Yucatan, and now in Tulane. We are collaborating with Eric moved to Tulane just to do the experiments with macaque. And fortunately, data to the diet of mice and dogs and reproducing on macaques, we are really very hopeful that the results in the clinical phase will be very exciting. Well, thank you very much. Well, before this is my institution that make my salary, and also CONACYT that pay the scholarship for my students, and also some of the support producing plasma at the beginning for the support of all of these years. Thank you very much for the attention.
Are there any questions? Dr. Jaime Ortega, yes.
Hello. Are these proteins recognized amascotes and trichomascotes or only trichomascotes?
I'm sorry. No.
Si las proteínas son de amastiones, ¿de qué?
Okay. Well, the TSA1 is also a trichomastodon. The TC24 is all of these stages.
Okay. I want to know whether there are some other examples of vaccines, commercially available vaccines, using recombinant.
Well, there is a commercial, there is a, for example, the hepatitis is a recombinant protein. What the flores that are produced is produced in the picture pastores. But there are other commercial, there are several. Even the companies, also is producing baculovirus.
Just following up with the question, do you think you can make an antivenom using recombinant proteins? Do you think that is possible? For example, an antivenom for jellyfish skin.
You use an antivenom, the recombinant protein for antivenom.
Yes.
It can be used. It can be produced antivenom for recombinant protein. But there is the IUD is Gerardo it's using the proteins, recombinant proteins to produce venom, and without depending on milking the scorpions or even the snakes. But especially that know that Gerardo also is really working hard because this protein from venom, especially from the scorpion, has a lot of systems and problems. One of the problems that we have in many proteins from the parasites, especially the protozoan parasites, they have a lot of problems and systems. So besides that, we are using the chapter on this enzymes that promote the folding in the parasites, or the same parasites, for example, in Trichomonas, the group of type that demonstrate that cyclophilins or bacterial proteins of parasites are very specific for some of the molecules in Trichomonas. Just the isomerization analysis probably wouldn't problem for the reforming of proteins to produce any product. Well, Pronomal is a pharmaceutical company. It's fantastic. This is the one that produced the prototype of the vaccine for clinical phase. Is interested in it was, I hope it's and will be the company that is.
So we have the last question before going forward. One more last.
Thank you. First, have you considered another kind of technology like mRNA or new technologies for the make your vaccine?
Okay. Well, I didn't show here because it's a new project in Texas Children's Hospital, and also there are new prototypes, some new antigens using TC24 and other antigens as RNA vaccines. They already tested it. They want to test it in Yucatan. You know what is the problem with more than 6 months getting the person to get the vaccine. That can be a huge problem. But yes, RNA, there is 2 or 3 papers already published using another vaccine for Chagas disease. Okay.
Now we really have the last one before.
You think using both protein to induce immune response in your patient, or you are going to produce either one of them?
Okay, well, you know, the funding sometimes are limited. So the lead foundation want to go as fast as possible to use the put vaccine already there. TC24 is a really, really good antigen. But the thing is, ESF1 has another problem. That is why we are looking to new alternatives to put it in 2 months. But you know, when you use a fine tool, you have to test the symphony. So that implies a lot of money. So it's use TC24 first, and we can keep working to integrate ESF1. In this case, we avoid to use the inclusion values in ESF1 because this new strategy on new epitopes. They already have 15 epitopes, new ones that nobody has used it. So we are going to test this.
Thank you.
So this is an elusive parasite that doesn't want to be fought with a vaccine, but I am glad that you are coming forward with that. So we have now, everybody can do whatever they want for a little while. Personally, I will go for lunch. But at 3 o'clock, there are the posters of our students that have come here. So I invite you all that you come from 3 to 4:30 to listen, learn, ask them so many questions as part of their training. So have a very nice